And good morning, micro peoples. So, adaptive immunity is what we're talking about now. And that, innate immunity, it's immunity and it is protection, but it's doing it by rather obvious methods. I mean, you could, the habit of washing your hands could potentially be considered innate immunity. These are things that you do to serve as barriers, and so the fact that our skin just does what it does makes it into a really good innate system. The flagella in your lungs that are constantly sweeping stuff out, another good way of maintaining innate immunity. And actually, just something that occurred to me is that people that have that struggle with cystic fibrosis, which is a defect in a chloride transporter, and specifically affecting the lungs, in that without the chloride transporter functioning properly, you end up osmotically generating a lot of really heavy, thick mucus, particularly deep in the lower lungs. And you have to have, there's a special technique for, sort of patting people on the back to loosen the mucus, coughing it up, oh boy. But the fallout of this is, because they have this heavy mucus, they're not able to push it out as well as a normal person does. They end up having way more upper respiratory infections. And so pneumonia becomes a real serious, real common issue. And this is an example of what innate immunity can do for you, and does for us all the time. But so the innate immunity is doing so much that we don't even think about. The fact that you have your skin is intact. I actually have a little boo-boo on my pinky, and I've been kind of keeping an eye on it, going, "Oops, should I put some antibiotic cream on it?" Because you start to break apart this skin layer, you have the opportunity for infection. All right, so then we have acquired immunity, and this is where we're talking about or adaptive immunity. And this is immunity that we can generate against very specific kinds of molecules. And the primary, or one of the primary components of adaptive immunity is the antibody. As we were mentioning before, antibodies are generated and differentiated in individual B cells in utero. And so you've got a B cell that is simply circulating around, very slowly dividing, and waiting for an antigen that will bind to the variable region of the protein sticking out of the cell surface. And in fact, it's a IgM. And once the antibody binds to an antigen, that triggers that B cell to start going into cell division. And so you begin amplifying up the cells that produce the antibody against a specific antigen. That cell was there, but it was maybe a single cell. But now, every day, you're dividing. And so the statement that you're not really going to have immunity the first day or immediately after the vaccination. And that's because you're generating a B cell population that produces an antibody that recognizes that antigen. The booster shot is simply just elaborating on that immune response. And as to why Johnson & Johnson COVID vaccine doesn't require a booster shot and Moderna and Pfizer do, I am embarrassed to say I had to look it up. I suspect it has to do specifically with how long-lived the antigen is in the body. And it has to do with the delivery system. I mean, that's really all the differences between those two guys. And so we also know that, I'll say this, but single dose is doing pretty good. Definitely get both doses. But a single dose is going to provide a significant level of immunity. But the second dose is really good because that is further amplifying and causing the class switch to actually start to really ramp up. And what you want to do is generate B cells that can produce all these different classes of antibody. Each one has the same variable region, but because they have different constant regions, it's going to be called class switching. Different B cells are producing some antibodies that are on the cell surface, some antibodies that are kind of complex together, and they are able to be able to precipitate out things like viruses. Some antibodies are floating around in the blood. These are really good at neutralizing antigens very quickly. You may have heard of something called a gamma globulin shot that is sometimes given for people that have been exposed to hepatitis. If you work in a OBGYN wing or area, you may have heard of something called a RhoGAM, RHOGAM. RhoGAM and the gamma globulin shot, what they're doing is actually injecting you with antibodies. And these antibodies give you temporary immunity. They'll be scavenging something up. So in the case of hepatitis, we're starting to generate hepatitis vaccines. But if you've been exposed potentially to hepatitis, and I know back 30 years ago, you'd go to the doctor and they would give you a gamma globulin shot. This is an antibody that is going to scavenge up the virus and prevent it from infecting things. And it works pretty well. With RhoGAM, if the baby is Rh positive and mom is Rh negative, that is the positive or negative in your blood type, you may be A positive or O positive or O negative. That positive or negative is something that's known as the rhesus factor, the Rh factor. And it's a protein that is on the cell surface of your blood cells. Regardless, you either have it or you don't. Now, if mom is Rh negative, but dad is Rh positive, the child could be Rh positive. And during the birthing process, there could be sufficient blood being generated and spilled. Oh, God, if you've ever seen a pregnancy, I don't want to talk about it. In that mom could get exposed to the baby's blood and begin generating antibodies against the Rh factor. Now, if all she's having is one baby, it doesn't make any difference. But if you have a second child, and that child is Rh positive as well, now you have a slight immunological issue of mom having antibodies against the Rh factor in the baby, and it can generate some anemia. And I don't know how critical it is, but it ain't good. I'm not sure how fatal it is. Regardless, what they do now is they scavenge up any of that Rh factor that mom might have been potentially exposed to by giving you a shot of the antibody collection. The antibody binds everything up, it's gone. It basically erases that protein from your system so the immune system never sees it. So antibodies then can do a whole range of things, and allowing them to class switch, allowing the B cells to differentiate, to generate all the different classes, just provides you that much better antibody protection. All right, so another part of the immune system, and I will tell you that A, immunology is fascinating, B, it is pretty darn complicated, and C, what we now know about immunology in the past 30 years is skyrocketing, because prior to HIV, immunology was not all that well established. But we know a lot about it, in part because of discussions of HIV and research done on people that have the virus that causes acquired immune deficiency syndrome. All right, so there are a class of proteins that are primarily on the cell surface called the major histocompatibility proteins, the major histocompatibility complex, MHC. When you hear about people talking about a tissue match, what they're measuring and looking at is how similar or non-similar individuals' MHC proteins are. And a good match is going to have a really close MHC. MHCs are also being generated by a similar kind of somatic recombination event that's happening with the production of the antibody variable region, in that you can have a bunch of different pieces and you pick one from this group, one from this group. Different parts of the gene get combined to create a whole range of MHC type proteins, and that's what determines self. So at least as long as your immune system is working properly, you are not going to begin establishing an attack against self. But this is one of the things when we start thinking about the immune system, is you're turning loose a class of proteins and cells that, not trivially referred to as killer cells. You're turning that loose, and you don't want it to begin attacking your own cells. You want it to begin attacking cells that have been infected by the virus or the bacteria. But, you turn a army loose, and hopefully it does its job, but doesn't go too far, and that's when we start to have autoimmune diseases. So MHC1 is on all cells, except it turns out it's not on red blood cells, which is wonderful because that allows for a very easy blood transfusion system. If MHCs were on blood cells, blood transfusions would be as difficult as a liver or kidney transplant, but they're not there. So red blood cells don't have major histocompatibility, which means that all you have to do is have the appropriate blood typing, and you're good to go. On the other hand, O neg is the universal, and so in an emergency, one of the first things they're doing is typing the blood and identifying the blood type. But if it's a real serious emergency, they just start going ahead and pumping you with O neg. Regardless, they're present on all cells, and these proteins are doing something called presentation. They are putting degraded antigens onto the cell surface, and they are presenting this to the lymphocytes, the white blood cells, and it essentially is, they got me. That is, I've been infected. So this is a warning that we've got some kind of a viral invader, and the virus gets into the cell. The viral protein is broken down by proteases. These small fragments are then associated with the MHC, and then the MHC is being dumped out onto the cell surface. And just as a little point, the Moderna and Pfizer vaccines are producing an RNA that gets into the cell and then translates this specific protein, and now we have the next step happening. So, another class of the major histocompatibility complex is MHC2. And this is also present on antigen-presenting cells. And this is different than all cells. Antigen-presenting cells are part of the primarily the bloodstream, but also in some dendrocytes, some neurons and things. And these are presenting the antigen to the lymphocytes specifically. And they are essentially training the B cells to start producing the appropriate antibody. Kind of like, look what we found. You need to go kill this. So we have the initial, they got me. That protein then is going to be presented on to the MHC2 on very specific kinds of cells. All right, so then the other part of this, we've talked a little bit about this, are the antibody-producing cells. These are known as the B cells. So B cells, they are making antibodies in response to the presence of the antigen. And the T cells are essentially directing this. This is also known, this part of adaptive immunity is known as humoral immunity because it is coming from the differentiating B cells that are found in the blood and the bone marrow. All right. So all B cells, when they begin life as a baby, actually as in utero, I'm going to turn on one set of lights. It's too dark in here, man. Make me crazy. All right. So The B cells, they underwent somatic recombination to produce the specific antibody. And the B cell is just hanging out. So when we think of it as just a naive B cell, it has undergone somatic recombination. It does have an antibody that it is sticking out there, but it hasn't bound antigen yet. It's waiting to be tapped and said, hey, you're up, we need you. When it does bind to its antigen, it begins to divide like crazy. It starts to expand what we think of as the clonal expansion. And some of these cells will actually become memory B cells. This is how you can respond to an antigen, an infectious agent, years after having it, after having been exposed to it for the first time. And so there are some systems between a vaccine and the B cell that is generated or that is activated this way that have real long-lived response. Others don't, and it just has to do with how the system is working. But memory B cells, they're able to, upon reinfection, begin immediately dividing and secreting out antibodies, and you've got a really effective immune response. So I've been inoculated against smallpox. You haven't. And so in the event of a smallpox outbreak, I have hopefully memory B cells that will provide me with an immune response. The truth is, I think that as long as it's been, my memory B cells probably aren't all that functional. Regardless, that is the way it does work. All right, so we've talked a little bit about this, that the B cells are generated randomly in utero. You just make a whole set, several million, B cells with different antibody variable regions on the external surface. And if they bind to an antigen, that causes them to, essentially, it's a autocrine signaling. That is, they secrete a hormone that binds to a receptor right on their cell surface that causes them to begin dividing more. And they begin dividing and dividing and dividing. What you then have is a bunch of cells of this proliferating clone. They begin producing identical antibodies. And a few of these do become memory cells. Later, maybe even years, if the memory cell encounters the antigen, it can divide very rapidly and begin to produce another clone that makes antibodies and some more memory cells, and you can maintain this immunological response. Now, we're going to just take a moment here, because those of you in TV land, those of you in class, how many of you have ever heard of a monoclonal antibody? No? Oh, all right. There is a COVID-19 treatment that is known as a monoclonal antibody treatment. Monoclonal antibodies are a really big deal in that it's a way that we can generate antibodies at essentially an industrial pharmaceutical level. And they kind of underscore the way the immune system is working and the way antibodies are being generated. And so it's worth going through a little bit. If you haven't heard of monoclonal antibodies, keep your eyes open for it, because you will see them around. There are whole class of some arthritis drugs, a lot of chemotherapies that will have, like, I'm just pulling this one out from the top of my head. It's not an actual one, I don't think, but something like this, Remifulab. If you ever see a drug that has the AB at the back end, and there's Remizabab, and all these different AB, these are monoclonal antibody treatments. They're going to be given IV primarily. They're pretty expensive, and when they work brilliantly. All right, so this is how we generate a monoclonal antibody. And it sort of underscores the whole antibody operation. You take an animal, typically it's a mouse, just because mouse monoclonals have been the initial system that has been developed and that works really well. There are other ways you can do it, but we're going to stick with the mouse right now. So you have a mouse, and a six-legged mouse. All right, you inject the mouse with a very small amount of antigen, using the mouse, and you allow him or her to begin making antibodies. What you're doing is the antigen is binding to the B cells the mouse has produced in utero. If the B cell can bind the antigen, it begins replicating. And you let this go, about 4 weeks or so. So you're generating an immune response. Unfortunately for our mouse, we're going to have to take its spleen, which I'm afraid to tell you is going to kill the mouse. Sorry. You flush the cells out of the spleen, and you can identify and culture B cells. You then take B cells and you fuse them with a tissue culture cell line. And this is getting beyond the scope, but tissue culture cell lines are cells that have undergone significant mutation such that they are immortalized and they divide all the time. In a sense, they're cancer cells. So you take B cells and using actually a little bit of like polyethylene glycol, essentially it's antifreeze, you can fuse cells, actually cause the membranes to fuse together. So you have a B cell and you fuse it with an immortalized tissue culture cell line. And there are specific culture cell lines that are used. And you then take this collection of cells, and again, I'm not sure if you've ever seen what's called a 96-well plate. It is a plate that's got about that big, and it's got a bunch of little wells in it, and we use them up the stairs all the time. 96-well plates were pretty much invented and developed for monoclonal antibody work. And what you're going to do is, through dilution, you're going to set up such that you got this plate with 96 wells, and I'm going to draw all 96. No. Each well has got a B cell in it. They also have a layer of what are called feeder cells that have been irradiated so they don't divide and don't contaminate things, but they're producing stuff. All right, so you have a little bit of tissue culture media here in that well. I'm drawing kind of small, but hey, you missed out. All right, what you then do is you take a little bit of this tissue culture media and you look to see, does it have an antibody in it? And there are a number of different assays you can do for this. You look to see if it binds antigen. And what you're doing is you're identifying a B cell that is producing the antibody against the antigen you made. And so it's a bit of a hunt and peck. You're just having to look to see which one produces the antibody. And you start looking through, and if you did a good initial immunization, you'll have, a number of cells that are producing antibody. And the cool thing is, because this has been fused to a tissue culture cell, you've got now a tissue culture cell line that produces a specific antibody. Now, when you injected that mouse, or when you get injected, there are a number of B cells that can bind the antigen. And so, as they begin to divide, you develop what is called a polyclonal, multiple B cells, a polyclonal antibody. That is probably the best for doing neutralizing reactions, is that you have you have a whole bunch of B cells. Each one is producing an antibody that binds to different spots on the virus, the antigen, whatever. But, it's something that you're having to do temporarily. With this case now, we have a cell line that produces a specific antibody, and this is now called a monoclonal antibody. And the really cool thing about monoclonals is that we have facilities in place where we can take a tissue culture cell line, grow it up in not bats, but pretty big tissue culture plates. A good monoclonal antibody that you select for, the cell line is secreting the antibody into the media. And so all you do is, at the end of every week, you suck the media off this big set of tissue culture plates. You have now media that contains the antibody to whatever, and you laugh to yourself because you have sucked off, probably $250,000 worth of pharmaceutical at the end of every week, probably like $5 million for this stuff. You can very easily purify the antibody, and that's when you then begin marketing. And as we mentioned, antibodies work outside of the cell. They work in different organisms. You inject this into some dude, the antibody binds the antigen, and it does its job. So by developing monoclonal antibodies against a whole range of different proteins, we can provide somebody with a little temporary immunity, a little bit like RhoGAM. So monoclonal antibodies are amazing, amazing Nobel Prize winning discoveries. All right, so the key point here is when you inject somebody with a monoclonal antibody, you're going to be doing IV, this is providing them with temporary immunity. It's not going to be an adaptive immunity or an immunity that they can acquire, because you're not giving them the antigen, you're giving the antibody. So now, in our adaptive immunity, we've got some things happening here. And wait for it. This is why, A, you have to wait a while after you've done the first injection to have an immune response, and B, why it's useful to have a booster shot, because it takes time for these cells to divide. So when you're making an antibody, you inject on day 1, and in about 2 weeks, you begin generating a fairly quite detectable, easily detectable amount of antibody. You begin to have class shifting, where you go from IgM to the secreted form that's now circulating around, the IgG. IgG is a really nice form of the antibody. That way, no matter where the port of entry is, the nose, you get jabbed, on your hands, on your eyes, you've got circulating antibody that can take this antigen out. So you begin to get the IgG shift, and now we're going to have the boost. Now, the boost interval can be really quite a while, but it can also be, a month later, you get your booster shot. And now you have the secondary response, and this is why it's so important to get the booster, is that the amount of immunoglobulin G producing B cells goes way up, and your IgG really goes up. So for sure, if you're going to be potentially exposed to something like COVID, and you want to have the best opportunity to actually not have any symptoms, having a booster is going to help a lot. So the primary response is pretty slow. The antibody production is a little low, a lot of IgM, and what this means is you're going to get sick and then you recover. The second time you see that immune response, though, the second time you are exposed to that virus, boom, you get a really strong antibody production in the secondary response. You're not going to have any symptoms, and you're going to be able to defeat this thing really fast. So you also now, once you've been exposed and allow the exposure to go, you begin generating all these other kinds of immunoglobulins. And so a number of things can happen from that as well. But let's just see here for a second. What's the next bit here? Yeah, all right, actually, we'll get you back to it in just a moment. Now, so a booster is a good idea. Now, another thing you may have heard, and this actually has to do with, I think, IgD. There are antibodies that are secreted in breast milk. And so this is a really interesting kind of response and opportunity during breastfeeding. And it goes like this. So when you were born, your intestinal epithelia has not sealed up very well. And you have the opportunity as a little baby, particularly up to about 6 months, to have material that is in the gut actually being put into the bloodstream. Now, hopefully, it's not going to be things like bacteria, not that big, that'd be really bad, but big proteins can get into the bloodstream. And a couple of things this falls from is that you really need to be very careful about what you're feeding a infant up until their gutty wets seal up, which is about 6 months or so, 4 to 6 months. Prior to that, their guts are pretty porous. And so what you're feeding them, some of the proteins actually are getting into the bloodstream. And if you're not careful, you can generate some weird allergic responses. But the cool thing is, when mom is feeding the baby breast milk, there are immunoglobulins in the breast milk. And these immunoglobulins are able to get out through these sort of leaky channels and into the bloodstream and provide the baby with temporary immunity. So, if you've had a baby or you ever have one or know people that do, one of the things you'll see is that for the first 6 months, 5-6 months of breastfeeding, the baby doesn't get a whole lot of colds. And primarily what's happening is as a cold virus is circulating around, mom is able to get it. The B cells she has produce antibody. They're being secreted into the breast milk because she probably has been exposed to this cold virus multiple times. And the baby now has temporary immunity. And once the gut seals up, once you start shifting from breast milk to solid foods and things, but mainly once the gut seals up, the baby can't get that temporary immunity anymore, and that's when your baby starts to get their own colds and things. And so, you'll have a period, a little window where you're doing a lot of breastfeeding, but happily, you don't have a lot of colds. And then all of a sudden, when you stop breastfeeding or when the guts seal up, more likely is what's happening, man, your kid's coming down with colds all the time, little petri dishes. All right. So that is what allows for the transference of immunity through breast milk. Okay, so a few more cells to talk about. These are the T lymphocytes. And these are some of the cells that the virus that causes AIDS actually knocks out. The helper T cells and the cytotoxic T cells, these are some of the guys that end up being destroyed by the virus that causes AIDS. So we have the lymphocyte, which is in the bone marrow. It can differentiate, and it differentiates into a cell that is generating a protein called CD4 or a cell that is generating a protein called CD8. CD4 cells, they become the class 2 MHC. CD8s become the class 1 MHC types. And these are then further able to interact with antigen. And you have different classes of T cells, that are known as the helper T cells, the regulatory T cells, and then cytotoxic T cells. And you don't need to be overly concerned about the different kinds of CD4 and CD8, just that we have classes of these different kinds of T cells. And the T lymphocytes, these are responsible for a whole set of cellular immunity responses. And so we have a protein or a cell that has been infected. And this is going to stimulate the interaction between the helper T cell and the cytotoxic T cells. And the helper T cells, one of the things they're doing is beginning to secrete different kinds of cytokines. Interleukin is one of them. These are inflammatory responses that are involved in cell lysis. They're part of what the reddening response around a wound is doing. The CD8 cells, they are also developing the ability to respond to altered cells, and they can also develop various kinds of memory cells as well. All right, so a couple of the things that the cytotoxic T cells can do is they can put in to the cell membrane of the offending cell a protein known as a perforin, which is perforating the cell membrane. It allows the attacked cell to be subject to all kinds of osmotic stress, and the other protein that gets put into it is something called granzymes, which are a set of different proteins, and these are causing that cell to go into programmed cell death. I think we talked about this, something that's known as apoptosis. Just so you know, the second P is silent. If you say apoptosis, you sound like a hick. That's my feeling, though. Apoptosis is a very specific way for a cell to die. I mean, obviously, if you take cells and lyse them or scrape them and sit them on the bench, they dry up. Cell death can be a very specific thing, and that there is, in fact, we now know, signals that you can trigger a cell to go into cell suicide. It's essentially like destroying the cell contents before the enemy can get a hold of it. Regardless, cells go into directed cell suicide, and this is a way you can program invading cells to go into cell death. And this is specifically important in that cytotoxic T cells are very good at identifying cancer cells. Now, these are cells that are self, but these are cells that have gone off the rails. They've gone off the reservation. They're dividing. They are going to be presenting some cues to the cytotoxic T cells that this is a cell that is not behaving properly, and while it is self, you're able to take it out. The lack of the cytotoxic T cells is what was one of the first tips that HIV, the virus that causes AIDS, was affecting T cells, in that people were beginning to show that folks that were infected with HIV were showing specific cancers that are typically phenomenally rare because our immune system is so good at taking them out. And we will pick up on some more T cells and issues on Wednesday, just so we have some things to fill out the class, and we'll be kind of filling things in from there. All right, ciao.